if there are no questions, then perhaps we could go on to the other speakers, and their topic is livestock fodder management and livestock-based livelihoods. I would like to invite Dr. Luming Ding, an associate professor at the Department of Ecological and Environmental Sciences, to give us her talk, please. Ms. Ding, are you there? Are you there, Ms. Ding?
Yeah, Dr. Ding is here.
Okay, great. I would like to.
Oh, sorry. Can you hear me?
Yes, we can. Thank you.
Sorry, my computer just was power off.
No problem.
Welcome.
Looking forward to hearing you.
Okay, I share my screen now. Can you look at my screen?
Yes, we could see it. Thank you.
Okay. Okay, I start. Firstly, thanks for this opportunity to share my lecture. My topic is on forage and livestock. More accurately, it's herbivore or ruminant, not livestock. I'm working in Lanzhou University in China. The contents, the following contents I will speak. Firstly, I start from stem of forage. The function of the forage is to support and connect other organs with the root, and conduit for water and nutrient transport. Stem also have some limited photosynthetic activity, like leaves, and the stem contain both actively growing or dividing regions and mature or fully differentiated regions. And the leaf, we move to leaf. The leaf structure, from the picture we can see the upper and the lower epidermis is heavily coated with waxy cutin. The cutin can conserve water and protect the leaf. There are two kinds of cells. First is palisade parenchyma in this part, and the second is spongy mesophyll. So from the picture, we can see the two kinds of cells are different. The first is neatly, tightly packed, and this spongy mesophyll is loosely packed. And the light reaction of photosynthesis in this palisade parenchyma cells, and the dark reaction or respiration in this cell, in these cells. In the photosynthesis, the carbohydrate should be produced and to move to different parts of the forage. During day, the leaf produces more sugar than can be transported to root. And also during day, the carbohydrates will move to root. The extra carbohydrates will form starch crystals. So the products of photosynthesis will be transformed to glucose or fructose and to produce sucrose to roots or as starch. And at night, the carbohydrates from chloroplast move to root. And after harvest of the forage, the starch will flow from root move to top for regrowth of the forage. The nitrogen fixation of the forage. The nitrogenous enzyme converts nitrogen to ammonium using carbohydrates as the energy source. Ammonium secreted to cytoplasm of the cell and converted to glutamine and glutamate. So these two can be converted to transport amino acids as aspartate or aspartame. Under drought stress, the forage will respond. So the morphological response can be found. The stem of the forage elongation reduced, and the ratio of leaf to stem increased. The yield of the forage declines, but the quality of the forage increases. So in the drought area, the quality of the forage is better. It is also the same for fruit or vegetable. And under drought stress, how to manage the forage? Of course, harvest is economic to get forage product. And move or clip of the forage to encourage the regrowth is also another choice. And the cold adaptation. So in cold conditions, the plants have evolved some mechanisms. Supercooling of the cell solution. It means cooling below 0 centigrade, the cell will be not freeze. The mechanism is accumulation of soluble sugars. Root and crown starches break down to produce sugars and reduce the amount of water lost during extracellular freezing. Another is stabilize larger molecules and membranes within the cells, and these functions can sustain the curve. There are some plant main compounds we should consider for the quality of forage: carbohydrates, nitrogenous compounds, and lignin. So there are some different carbohydrates. First is non-structural carbohydrates, NSC. There are 3 kinds of NSC: monosaccharides, disaccharides, polysaccharides. So they play different roles. The monosaccharides is a kind of basic units of the carbohydrates. Disaccharides play a transporting function. Polysaccharides is a storage function. The monosaccharides, glucose and fructose are the monosaccharides. Sucrose is disaccharides, which can be made by the combination of glucose and fructose. Polysaccharides, like starch, it's a kind of storage carbohydrates. The second is structural carbohydrates, like pectin, hemicellulose, and cellulose. These structural carbohydrates are polymers. So pectin is a polymers or alpha D, glycuronic acid, and hemicellulose is the polymer of xylose and mannose. Cellulose is the polymer of glucose. Sorry, my computer cannot move again.
Oh, he's
I'm sorry for the so many problems. Okay, it works. Now we move to lignin. It's a kind of polymer of phenyl propan units. The function of lignin is glue binding cell wall together, and water impermeable, undigestible. So if the higher contents of lignin in the forage means lower quality of forage. The nitrogenous compounds include ammonia, ammonium, and amino acids, protein, DNA, and RNA. Another part is energy. So energy play a role for maintenance needs. Maintenance needs include basal metabolism, energy requirement, higher for extra activity, like grazing animal will need much more maintenance energy, higher in the cold environment. There is also energy for gain or for lactation. Energy can be defined the followings. Gross energy, it's a total energy in a feed ingredient, can be determined by burning all of a sample and measuring the heat given off per unit weight. Okay. Digestible energy is the gross energy minus the energy in the feces. There's another definition, TDN, it's total digestible nutrients. It's equal digestible protein plus digestible crude fiber, digestible nitrogen for extract, and plus 2.25 multiplied digestible other extract. So the TDN can be calculated. Metabolism energy, it's digestible energy minus the energy in methane from the fermentation and the urine. Fuel feeds are measured for metabolism energy. Forages usually have higher methane and urinary losses than feeds with low concentrations of fiber. Net energy, it's a metabolism energy minus heat of metabolic reactions not used for maintenance. So there is two kinds of net energy. NEM, it means net energy for maintenance, and NEG, net energy for gain. The same for with ME. Fuel feeds are measured for net energy. It is very difficult to measure, and the net energy are usually predicted from the total digestible nutrients or digestible energy. This diagram can explain the partitioning of energy. the growth energy, digestible energy, metabolizable energy, and net energy for maintenance, for growth, and for lactation, as explained before. Now we move to the second part. It's still the first part. The forage, we know it's used for livestock. So we need to identify quality traits that improve animal performance. Also the traits that reduce feed supplements cost. So there are some definitions we need to explain for the forage. Potential intake. So how much is voluntarily consumed by the livestock? Digestibility is how much is absorbed into the animal's body through the digestive system. The efficiency of forage means the ratio of animal production to nutrient supply. And anti-quality factors is the components of the feed that inhibit intake, digestibility, or efficiency. So we need to examine these items to determine the quality of the forage. There should be a list of analytical tests should be done to check the nutrients of the forage. It includes dry matter, moisture, crude protein, neutral detergent fiber, acid detergent fiber, neutral detergent fiber digestibility, calcium, and phosphorus, and ash. Dry matter we know is the percentage of feed that is not water. So moisture content is plus dry matter equal 100. The crude protein is determined by measuring total nitrogen in a forage sample and multiplying by 6.25. It is a mix of true protein and non-protein nitrogen. Neutral detergent fiber, NDF, important item, can be determined by washing the forage with solution, with NDF solution. The soluble cell solubles can be released, and the reserved is NDF. It has cellulose, hemicellulose, lignin, acid insoluble ash, Q10. So these are NDF. Neutral detergent fiber is the percentage of fiber in a forage sample which is not soluble in a neutral detergent solution. It is the residue left after boiling in neutral detergent solution. It is called ANDF if amylase and sodium sulfate are used during the extraction. So NDF represents cell walls, partially digestible because it contains hemicellulose. And NDF is inversely related to voluntary intake. Therefore, higher NDF content means lower palatability of the forage. Acid detergent fiber is similar detected as the NDF, also by washing. So cellulose, lignin, silica, and Q10, these are ADF. Cell solubles and hemicellulose are washed out. ADF and NDF, the difference is the hemicellulose. So hemicellulose belongs to NDF.
Indeed.
Acid detergent fiber is the percentage of fiber in a forage sample which is insoluble in a weak acid. It is the residue remaining after boiling forage sample in acid detergent solution. ADF represents cell wall minus hemicellulose. It is inversely related to digestibility. So therefore, higher NDF means lower quality or low nutrition of forage. Acid detergent fiber. So ADIN greater than 15% of nitrogen is an indication of heat damage of forage. Formation of ADIN is also called non-enzymatic browning or Maillard reaction. The neutral detergent fiber digestibility is a portion of neutral detergent fiber lost during incubation with rumen fluid. The minerals of calcium and phosphorus also commonly analyzed in forage to estimate the ratio of calcium to phosphorus. So generally, this ratio is good for feed or forage. Ash, we also called total ash or crude ash, is an estimate of the total mineral content, the residue remaining after burning a sample. Ash values about 8% for grasses or 10% for legumes usually indicate soil contamination of the forage. Each 1% of soil contamination is 1% less of total digestible nutrients of forage. So ash, ADF ash, and NDF ash will be different because ADF and NDF procedures remove some minerals.
Okay.
There are still some calculated terms in forage testing. The previous are tested in lab, but these are calculated, such as adjusted crude protein, digestible dry matter intake, net energy for lactation, relative feed value, relative forage quality. Adjusted crude protein is a calculated value adjusting total crude protein for heat damaged protein. Adjusted crude protein estimates protein available for animal use. It should be used to formulate diets when the ADFIN is greater than 15% of the nitrogen of sample. So it means much more crude protein cannot be used by animal. So we should use adjusted crude protein. Digestible dry matter, DDM, is the portion of in the forage that is digested by livestock at a specified level of intake. It is also called in vivo digestible dry matter if determined by feeding animals in a digestion trial. It is often estimated by measuring in vitro digestibility, in situ digestibility, and it can be calculated from the ADF content by the following formula. Dry matter intake is a very popular used definition. It is an estimate of the relative amount of forage an animal will eat when only forage is fed. So it is very difficult to measure for grazing livestock. Okay. Net energy for lactation, NEL, is expressed as megacalories per kilogram of feed dry matter, which can be calculated through total digestible nutrients by the following formula with the TDN. Relative feed value is an index which ranks legume and legume grass forages by digestible dry matter intake potential. So RFV index can be calculated by digestible dry matter and the Trimetry Intake. Now we move to the second part, forage products, hay and silage. We know after harvesting of the fresh forage, we need to dry it to make silage or hay. When the water content is lower than 20%, it is good for making hay. But from 50 to 20% of the water content normally used for silage.
Okay.
If the water content of the forage is higher than 70%, so much more effluent will occur. But if lower than 50%, the silage will have a risk to be burnt or heated. So hay vs silage. Hay need large field but small storage loss. Silage conversely need small field but a large storage loss. When we preserve the hay, we need to control the mold or inhibit the growth of mold. Molds grew at 20 to 35% moisture. The molds can consume nutrients such as sugars or starch, and the respiration of mold causes heating. So sometimes incur the fires of hay. Also produce toxins, which is detrimental to animal health and decrease food intake. Also produce spores, if inhaled by livestock, can cause lung disease. If the forage has mold, will reduce the value of hay. So we can use some hay additives to avoid the growth of mold. Drying agents can enhance the field drying. Also preservatives or inhibitors, it can increase moisture range for safe storage. There are some list of drying agents or inhibitors normally used. From the figure we can see, if we use the drying agent, the drying time in the field is shorter than without using for alfalfa hay. The hay production from the moment of the forage is cut until it is fed to the animal. Biological and mechanical changes occur that decreases the quantity and the nutritional quality of feed. Therefore, the goal to make quality hay is to conserve the protein and energy in the forage and maintain the protein in a form that can be effectively utilized by the ruminant. And the silage, during produce the silage, it needs to exclude oxygen from the silage mass, and the lactic acid producing bacteria can be stimulated, and the other molds or yeasts can be inhibited. The pH will be reduced rapidly through the bacterial growth and fermentation, and minimize the respiration losses. Lactic acid fermentation requires sufficient plant sugars, lactic acid bacteria on forage, anaerobic conditions, and proper dry matter content. The figure shows the several stages of the silage when making silage. The first stage, aerobic, there's still some oxygen. And the second is anaerobic, the pH decrease and the LD increase. But in the third stage, it is stable. When feeding out, the oxygen can re-enter and there is a second fermentation. Therefore, there are some losses of silage. So the first stage, aerobic phase, oxygen trapped in the air, species of silage. is consumed by plant respiration and aerobic microbes. The respiration process is most detrimental to silage quality. So loss of dry matter reduce plant sugar content. Also prolonged actions allow yeast and molds to grow. Heat produced and increase of the temperature of silage. There is still lag phase, fermentation phase, stable phase, feed out phase. I need to speed up. So there are some pictures of to making silage, a tower and a bunker and some bags or tanks. We move to the forage and the livestock. We know as a ruminant there are 4 stomachs: rumen, reticulum, omasum, and abomasum. The fate of the ingested forage at the beginning to floating in the on the top of the rumen, and then there is some rumen gas, mainly CO2 and CH4. And after some time, the forage will deposit to the bottom, and by ruminating to masticate it to smaller size and move to the reticulum. When the size is much smaller enough, it can escape reticulum to omasum and to abomasum. So there are still some factors affecting animal performance. So animal performance is the cumulative result of numerous interactions between the plant and animal. So potential performance will be affected by animal genotype, physiological status, animal health, and environmental effects. Forage quality is a function of nutritive value, intake, and anti-quality factors such as tannins, which will be affected by species, variety of forage, maturity of forage, and season, weather, or soil type. There's a part to share the grazing yak as a case. So yak, it is a unique animal born in Qinghai Tibetan Plateau as a rotational grazing. So there are some different grazing systems: 2-season rotational system and 3-seasonal rotational system. To combine some different seasonal pastures. The summer pasture, autumn pasture, and the winter spring pasture are now is much common in the yak grazing system. So the yak has some adaptive characters to resistance cold, adapt to low atmospheric oxygen and high solar radiation. Also adapt to harsh sward conditions like a short growing period of forage and short sward height for them to graze to survive. So, but there are some products of yak: milk, meat, fiber, hide, pack, and feces. Also, the yak also play an important role for cultural heritage. The
Dr. Ding, sorry to interrupt.
Okay.
You have a couple of minutes more, please, to wind up.
Okay, I will 2 minutes. Okay.
Thank you.
The forage biomass on Qinghai Tibetan Plateau, because the green period from June to September, so their biomass higher. Therefore, the quality, you see the crude protein, metabolism energy, and the digestible dry matter is higher in the grass growing period, but the NDF or ADF are lower in this period. So from the number of mastications or probolars, we can see the mastications are higher in the forage yellow drying period because of the higher content of fiber. You know, as the previous lecture, the stocking rate is very important for the management, greens management. So we need to have some optimum stocking rate to get the best utilized utilization of the forage. But normally we face much higher stocking rate. The stocking rate is the number of animals per unit area, and intake rate multiply stocking rate equal consumption mass per unit area per time. Therefore, we need to build some simulation model to help manage of the system. So the output is very easy for the manager to see and to get their decisions, such as at a different stocking rate and different grazing time to get the higher herd intake. So this model is from Israel Noamir model, this output. Okay, I have to say thanks again. It's my presentation.
Thank you, Dr. Ding, for such a detailed presentation about the importance of fodder and the kind of research that goes in to improve fodder quality, to increase stocking rates of animals in different ecosystems. I'd like to point out here that from the previous speaker, when Sergio was talking about the ecosystems and grazing in ecosystems, and it was nice to see how Dr. Ding we went from the ecosystem concept to the cellular concept, where he talked about cells in fodder, what total digestible fiber is, and it was very interesting. So we've gone from a large perspective to a very micro perspective, and he ended up with talking about stocking rates and what kind of management practices need to be followed to have adequate stocking rates of pasture animals. Thank you very much, Dr. Ding.